In the second half coverage of Chapter 9 on muscle physiology, we cover the single fiber muscle mechanics when the muscle fiber is stimulated. At the end of this video, you are expected to be able to discuss the two forces that determine the type of muscle contraction, describe the stages of simple twitch muscle contraction, differentiate between isometric and isotonic contractions, outline the relationship between load and velocity of muscle shortening, illustrate the relationship of stimulus frequency and the tension developed by the muscle fiber, discuss the relation between muscle length and tension development, outline the source of ATP and energy for muscle contraction, differentiate between three specific muscle fiber types in the human body and their basis of classification. In the mechanics of single fiber contraction, there are two opposing forces. The force exerted by a contracting muscle on an object is known as muscle tension, and the force exerted by an object on the muscle, usually its weight, is the load. Muscle tension and load are opposing forces. Whether a fiber shortens depends on the relative magnitudes of the tension and the load. For muscle fibers to shorten and thereby move a load, muscle tension must be greater than the opposing load. In the laboratory, muscle contraction can be demonstrated by a recording device that shows the event of muscle contraction as a parabolic tracing. A single muscle contraction of a single muscle fiber by a single electrical stimulus can be demonstrated by a curved graph called twitch contraction. The graph of the muscle contraction typically shows three stages of fiber contraction. This includes the latent period, and then the contraction period, and then the relaxation period. The latent period is a brief interval of about 1 to 2 milliseconds of a seeming inactivity of the muscle fiber between the application of the stimulus to the development of tension in the muscle. The latent period corresponds to the time of excitation events that occurs in the muscle prior to tension building. Then this period is followed by a steep upward curve called period of contraction from the point of tension development to the point of peak tension development in the muscle. The peak curve tracing of the contraction period is then followed by a downward curve called the period of relaxation, when the muscle resumes its original length. The tracing on the twitch contraction may appear as a very simple, possibly skewed kind of statistical curve, but there are very specific molecular events in these periods of muscle fiber contraction that underlies the simple line tracing. There are two main kinds of muscle contractions that can be exhibited based on the relative magnitudes of load and tension in the muscle. When the load is greater than the tension developed by a muscle, it produces an isometric contraction. In isometric contraction, the muscle develops tension but does not shorten and move the load or object. When the tension developed by the muscle is greater than the load imposed on the muscle, the contraction produced is an isotonic contraction. In isotonic contraction, the load or object is moved by the muscle. Comparing between isometric and isotonic twitches in the same muscle fiber, you can see in this slide that the latent period in the isotonic twitch is longer than that in the isometric twitch. However, the duration of the mechanical event, that is the shortening, is briefer in an isotonic twitch than in the isometric twitch. The reason for these differences is most easily explained by the following. In the isometric twitch, the tension begins to increase as soon as the first cross bridges attach. So the latent period is due only to the excitation-contraction coupling delay. By contrast, in the isometric twitch tracing or experiment, the latent period includes both the time of excitation-contraction coupling delay and a brief period of isometric contraction during which the muscle must develop enough cross-bridge cycling to enable the fiber to lift the load from the surface of an object. Similarly, at the end of the twitch, the isotonic load comes back to rest on a platform well before all of the cross bridges have detached. So there is another brief period of isometric contraction. The figure shown here clarifies the relationship of the load to that of the velocity of shortening of the muscle. So this figure shows the load-velocity relation. The velocity at which a muscle fiber shortens decreases with increasing load, and the shortening velocity is highest when there is no load, and zero when there is the load is equal to the maximal isometric tension the muscle can develop. But there's supposedly a third type of contraction that is apparent here. We call this as lengthening contraction. When the load is much greater than what the maximum isometric tension can develop, and so the muscle instead stretches because of the greater load than the maximum isometric tension. So you have lengthening contraction that is shown here. So all in all, you have three basic types of muscle contractions: the isotonic muscle contraction, the isometric muscle contractions, and the lengthening muscle contraction. The lengthening muscle contraction is sometimes called by other authors as eccentric contraction. Because a single action potential in a skeletal muscle fiber lasts only for 1 to 2 milliseconds, and the twitch it produces may last for 100 milliseconds or more, it is possible to stimulate the muscle to contract before its first contraction has finished. This event is called summation. As is shown here in the top figures, the isometric twitch following the first stimulus lasts for about 150 milliseconds. And when a second stimulus, S2, is applied 200 milliseconds after the first stimulus, you produce an identical twitch. When a stimulus is applied before a fiber has completely relaxed from a twitch, it induces a contractile response with a peak tension greater than that produced in a single twitch, or than the peak contraction by the first stimulus, and in this case S2. And this is a form of summated contraction or summation. But in this case, the summation is incomplete because we can still distinguish between the two contractions by the two different stimuli. But if we further shorten the interval of the stimulus, as in the case of S5 and S6, we produce a resulting peak tension that is even greater and smooth throughout. This is a summation of peak contractions that are complete. Now at the lower figures here, you have a simple twitch that is shown by the application of a single stimulus. But if we apply several stimuli, like 10 stimuli per second on the muscle fiber, we produce incomplete summation of peak contractions or unfused tetanus. But if you notice here at the first peak of contractions, you have a progressively increasing magnitude of contractions. This phenomenon is called tripe, which is the fundamental reason why we have to warm up when we engage in muscular activity. That is because in the initial contractions, when we initiate contractions of our muscle, we actually make calcium ions more available, and at the same time we increase the number of myofibrils that are contracting at increasing stimuli. So we have tripe contractions, which is a German term for ladder-like contractions. And here we have application of 100 stimuli per second. So in this case, we have fused or complete summation, fused tetanus, that produces a plateau of contraction. But this fused tetanus or summations does not go on indefinitely. There is a moment when these contractions begin to fail and produce a downward slope. At that point, the muscle will have already undergone fatigue, and continued application of the stimulus wouldn't produce contractions anymore in a fatigued muscle. And one of the primary reasons of this situation when you have fatigue of the muscle is because of the loss of energy reserves or source like ATP for the muscle to continue contracting. The figure shown here is a graph showing the relationship between the length of the muscle fiber and the tension that it can develop. The strength or tension that can be developed by a muscle may be changed by the length of the fiber before contraction. When a muscle fiber length is 60% of the optimum length or shorter, the fiber develops no tension when stimulated. As the length is increased from this point, the active isometric tension at each length is increased up to a maximum at optimum length. Optimum length is the length at which the fiber develops the greatest isometric active tension. Further increasing the length or lengthening the muscle leads to a decrease in active tension. At lengths of 175% of the LO or greater, the fiber fails to develop tension again. Although the passive elastic tension would be quite high when stretched to this extent, no tension can be developed beyond 175% of the length. The explanation to this length-tension relationship lies on the relative position of the thick and thin filaments of the sarcomeres. Shortening, or rather stretching a relaxed muscle fiber pulls the thin filaments past the thick filaments, changing the amount of overlap between them. Stretching a fiber to 175% of the LO pulls the filaments apart to the point where there is no overlap and no cross-bridge cycling can happen because of the distance between the cross-bridge or myosin heads and actin. At this point, there can be no cross-bridge binding of actin, of myosin rather to actin, and no development of tension. The tension decline at lengths less than LO or optimum length, which is the result of several factors. For example, the overlapping of the sets of thin filaments from opposite ends of the sarcomere may interfere with cross-bridges' ability to bind and exert force. And at a very short length, the Z-lines collide with the ends of the relatively rigid thick filaments, creating an internal resistance to sarcomere shortening. No other cell type in the human body is the rate of ATP breakdown that can turn as rapidly from one moment to another as in muscle cells. If a fiber is to sustain contractile activity, metabolism must produce molecules of ATP as rapidly as they can break it down during the contractile process. There are three ways a muscle fiber can form ATP. One is by phosphorylation of creatinine phosphate, which are small molecules produced from 3 amino acids and capable of functioning as a phosphate donor. The second is oxidative phosphorylation from the mitochondria. And then thirdly, the phosphorylation of ADP by glycolytic pathway in the cytosol. So you have three sources of ATP that can produce ATP for muscular contraction in cross-bridge cycling. You have creatinine phosphate pathway, and then you have oxidative phosphorylation pathway and glycolytic pathway. Muscle fiber identification or differentiation is based on two categories. You have maximal velocities of the shortening of the muscle and the ATP synthesis pathway used by the muscle to power the contractions. In the category of maximal velocities of shortening, you have muscles can be classified as either fast fibers or slow fibers. Fast fibers have high ATPase myosin activity at their myosin heads, and slow fibers have low ATPase activity at their myosin heads. And under ATP synthesis pathway, you can also classify muscles as either oxidative fibers or glycolytic fibers. Oxidative fibers rely on ATP production by oxidative phosphorylation in the mitochondria. So you have lots of mitochondria in these kinds of fibers, and they are surrounded heavily by numerous blood vessels. And you have lots of oxygen binding myoglobin in the muscle tissue itself. And finally, the appearance of these muscles are red. So we call them red muscles, mainly because of the preponderance of numerous blood vessels and the bright red myoglobin molecules when bound with oxygen. Among the glycolytic fibers, these are fibers that rely mainly on glycolytic pathway or glycolysis. You have few mitochondria found in these kinds of muscle cells, but with very high concentration of glycolytic enzymes. There's very little blood vessels around the glycolytic fibers because they don't need it, and a lack of myoglobin. So most often in appearance, these glycolytic fibers are whitish or white in color. So we often refer to glycolytic fibers as white fibers, whereas oxidative fibers are red fibers. Now, based on these two classification categories, we can therefore identify three types of skeletal muscles in the body. We have slow oxidative fibers or type 1. We have fast oxidative glycolytic fibers, so-called as type A. And then we have fast glycolytic fibers, designated as type 2X instead of 2B. Type 2B muscles are found mainly among other animals, especially mammals, except for humans. We have the type 2B genes in our bodies or in the muscles, but these are not expressed in the human body. To further differentiate between the three types of skeletal muscles we just identified, that namely the slow oxidative fibers type 1, the fast oxidative fibers type 2A, and the fast glycolytic fibers type 2X, we can further differentiate them based on the ATPase action and ATP source, the relative diameter of the fiber, and their fatigue susceptibility. Now, the slow oxidative fibers, or type 1, based on ATPase action and ATP source, you have low myosin ATPase activity in these fibers with high oxidative capacity. They have small diameters and therefore are thin muscle fibers, and they are highly resistant to fatigue because of their high oxidative capacity. They have lots of ATP. And then you have fast oxidative fibers with high myosin ATPase activity. Therefore, they are very fast muscles with high oxidative capacity. They have small diameters and therefore are also thin muscle fibers with medium resistance to fatigue susceptibility. Again, because of high oxidative capacity with lots of ATP available. And then you have the fast glycolytic fibers, or type 2X, which have a high myosin ATPase activity with high glycolytic capacity. And glycolysis only yields very little ATP compared to oxidative pathways. These types of muscles are large diameter, and so they are thick muscle fibers. And they are highly susceptible to fatigue because of their being glycolytic in ATP production pathway.